So first, I would like to thank Patrick for the invitation. So I'm an assistant professor at the University of Paris, and since the beginning of January, I have been working as a guest researcher at ETH Statistics. And so today I would like to present a work entitled l1-spectral clustering, a spectral clustering method using l1-regularization, which was done by in collaboration with Magali Champion, who is also my sister. So it's a kind of COVID home office work. Mélanie Blazère and the PhD advisors of Camille, Rémi Burcelin, who is a researcher at the French National Institute of Health, and Jean-Michel Loubes, who is a researcher at the Institut Mathématique de Toulon. Okay. So I will begin by a very brief introduction. It's more to give you some biological motivations of this work. So I don't know what your background in biology is, so I will begin by the very beginning. So genes carry the genetic information which is needed for the development of any organism. So if some genes will directly act to express a genetic trait of an organism, some other genes won't directly act on these organisms, but will activate or inhibit, so regulate the quantity of proteins produced by other genes. So all these genes are in interaction, and the interactions are represented by gene regulating networks. So a gene regulating network is a graph where one node corresponds to one gene, and an edge between two genes represents a relationship between genes. So if you have a directed edge, it means that you have a regulation between genes, activation or inhibition. And if you have an undirected edge, it means that you just have an interaction between genes. So when you work with gene-regulated networks, you have two different main goals. The first one is how using gene expression data, which give an idea of how many proteins are produced by all these genes. You can recover or infer the gene-regulated networks that models interactions between these genes. So from a biomedical point of view, if you know this kind of network, then, for example, the doctors can find a treatment which can block any interaction. And the second main goal is how you can explore the gene-regulated network to discover, for example, groups of connected genes and understand the biological diseases. So the associated keywords are the high dimension. So of course, when you work with biological data, you have around 1,000 of genes. And depending on the disease, you have around 100 of samples. So you have to develop some methods which can work with this kind of high dimensional data. The second important keyword is sparsity. So first, you would like to find the main interactions in the graph. So you want to recover a sparse graph. And then you have graph inference and clustering. So graph inference corresponds to the first point here, and clustering corresponds to the second point you want to explore the network. So there is another very important keyword I won't talk about in this talk. It's the causality. So of course, what you really want to do is to find directed edges. So you want to give a causal meaning to these edges. But here, we will not talk about causality. So when I speak about gene-regulated network, it's not a gene-regulated network, it's more a gene interaction network. So we don't care about the direction of the edges. So the main, so in this work, this work is more about clustering graphs, but I just would like to give you an idea of how you can infer such a network. So here we assume that we have some gene expression data. So it's a big matrix where you have all your nodes in columns, so all your genes in columns. And you assume that you have, you observe n replicates of all these genes. So you have a big matrix X. And you assume your data are drawn according to a Gaussian law with the mean 0 and covariance matrix sigma. So what you can first do to build an interaction network, you can say, okay, I have an edge between node I and node J. If the correlation between a node XY and XJ is significantly larger than 0. So you compute the correlation matrix, and given a threshold, you threshold your matrix, and you build the associated network by drawing an edge whenever correlation is significantly larger than 0. So fortunately, you can establish a link between the non-zero coefficient of this matrix, of this precision matrix sigma, and the coefficients derived from the linear regression of all genes on each gene. So here we just assume that each gene is written as a linear combination of all other genes except itself. So we won't, so we exclude self-loops. And so you can use whatever method you want, linear sparse regression methods, as it's well explained in the paper from Mychosen and Buhlmann. You can use any linear regression methods to recover such a graph. So whenever you find a non-zero coefficient, regression coefficient here, it means that you have an interaction between node xj and xy. So it works quite well. So from now on, we will assume that we know such a network. And I turn to the main part of this talk. What we really like to do is to find to use some clustering, or to define some clustering methods to first cluster vertices into groups that are densely connected and share a few links with the other groups. So it's more from a graphical point of view. And from a biological point of view, our objective is to discover groups of genes with similar characteristics to better understand a disease. So there are a large number of methods, of clustering methods. So you probably know a lot of them, So for example, you have partitioning algorithms, for example, k-means, which classifies nodes into a predefined number of groups based on similarity measure. You can also use hierarchical clustering algorithms, which build a hierarchy of clusters. And the last class of methods is spectral clustering algorithms, which use the spectral properties of the graph to recover the graph structure. So here I will begin by introducing the spectral as explained in the paper from Luxembourg, before presenting our new version of spectral clustering. So first, spectral clustering. So I need to introduce lots of notations of graph notations first. So we will, okay, so a graph J is just a set of vertices. So here we assume that we have P vertices corresponding to the P genes of the data set. And we have a set of edges, which are just pair of nodes. First, adjacency matrix. So the adjacency matrix is no more than a P times P matrix. It's a binary matrix, so you have a 1 where when you have a node, an edge between node I and node J, and 0 otherwise. So it's a 1-0 matrix. We also need to define the degree of any vertex. So the degree is the number of edges incident to I. So for each node, you just compute the number of nodes which are adjacent to I. And the degree corresponds to the sum of each column of the adjacency matrix. And we will denote by capital D. So capital D will be the associated degree matrix. So just a matrix where you have as diagonal elements, you have all your degrees and 0 otherwise. And finally, I want to define what a connected component is. So connected component is a set or subset of nodes from G, which are fully connected. So you have, I mean, all nodes are connected by an edge, and there is no connection outside the connected component. And a key partition C1 CK of G will be of J, G. Key partition C1 CK of G will be a set of connected components such that, so all these sets are non-empty. You don't have the intersection is empty, and the union of all these connected components form the entire set of vertices. Okay, so now I have some picture to show you how the graph looks like. So here we assume that we have an ideal graph. So this graph is made of k connected components. So in terms of adjacency matrix, the adjacency matrix looks like something like this. So it's a block diagonal matrix. So each of these block corresponds to one connected component. So you don't have any set loop. So you have a zero diagonal, and all nodes in a component are connected, and you have zero otherwise. So your adjacency matrix looks like something like this. And the associated graph, of course, something like this. Okay, so your graph is made of connected components. So now we assume that we observe a perturbation of this graph. So I define two probabilities of two perturbations. The first one is a probability pain of removing an edge inside a component, and payout is a probability of adding an edge between components. So we assume that we observe a perturbation of the previous graph. So here we didn't remove some edges, but we added some edges between components. So you have lots of edges, supplementary edges. And your adjacency matrix look like something like this. So you can still see this kind of block diagonal matrix, but you have lots of, I mean, it's a little bit messy. So you have some edges which are added or removed. So now if you want to apply the spectral clustering algorithm on this perturbed graph, you need to define first the Laplacian matrix. So the Laplacian matrix is defined as the difference between the degree matrix and the adjacency matrix associated to the graph. And the spectral clustering is based on the very nice properties of this Laplacian matrix. So first, you can show that L is symmetric and positive semi-definite. You can show that L has P non-negative real valued eigenvalues, which will be denoted lambda 1, lambda p. And the smallest eigenvalue of L is 0. Okay, so it's not so hard to see, to show this. And now, the property which is mainly used by the spectral clustering algorithm is the following one. The eigenvalue 0 of L is of multiplicity k, the number of connected components, and the associated eigenvectors correspond to the indicator vectors of the k components. So it's very hard for me to write something on my slide, so I will just explain quickly what an indicator vector is. So it's just a vector made of lots of 0 and 1. You have a 1 whenever when your node belongs to the component, and you have 0 otherwise. So now if you want to find, given a graph, you want to recover the cluster structure of the graph, you just need to do a spectral decomposition. So you do a spectral decomposition of L. So you will find 0 is an eigenvalue. The multiplicity of the eigenvalues, the number of connected components. And if you want to recover the structure of the graph, you just need to find the eigenvector associated to the eigenvalue 0, and it will give you directly the structure of the graph. So of course, this only works with non-perturbed version of the graph, which is not so useful, because if you have your graph is not perturbed, you can just see the structure of the graph. But you can imagine that if your perturbation is not so big, the eigenvalue will not be 0 anymore, but the smallest eigenvalues will be quite close to 0. So we can imagine that they will be very close, like for example, 0.01, something like this. So you can still do your spectral decomposition and see the number of eigenvalues which are close to 0, which give you an idea of how what the number of connected components is. And you can also imagine that your associated eigenvectors will be quite close to the true structure of the graph. So I have some other slides to better explain how it works. So the spectral clustering algorithm, as described by Loubes, is the following one. So as input, you have your graph G, you have the associated agency matrix, and the number of clusters to build, even if you can still doing spectral decomposition, you can still easily estimate this number of clusters. So first you compute the Laplacian matrix, defined as the difference between D and A. You do your spectral decomposition of L, and you store the eigenvectors associated to the first k at smallest eigenvalues. And you do a k-means just to recombine these eigenvectors and find the structure of the graph. So it looks like something like this. So in the ideal case, where your adjacency matrix is perfect and your graph is made of connected components, you do your spectral decomposition here. You find that the eigenvalue 0 is of multiplicity k. You derive the associated eigenvectors here, and you find the structure of the graph just because it's perfect, it works well, everything is already good. Now, when the graph is perturbed, you can do exactly the same. You compute the Laplacian matrix, you still compute, you still do your spectral decomposition, and you will, I hope we will find some eigenvalues close to 0 if the graph is not so perturbed. You compute the associated eigenvectors here. And if you do k-means, then it will reorder all nodes and you will still find the structure of the graph. So the spectral clustering algorithm is very famous for graph clustering because of its powerful computational results. You have also very nice theoretical convergence results in case of, if I remember, it's just for stochastic block models. You also have some refinements using other versions of the Laplacian matrix. But something, I mean, quite bad is that you have high sensitivity and no guarantee of recovering the true components in case of large perturbations, which is mainly due to the presence of these k-means. So based on this, we developed a new algorithm called l1-spectral clustering, where the Laplacian matrix is replaced by the adjacency matrix, so just for computational reasons. But the k-means is replaced by selecting relevant eigenvectors, which are written as solutions of specific L1 minimization problems. So now how it works. So we just, so as I said before, from now on, we will not focus on the Laplacian matrix anymore, but on the adjacency matrix, but it's exactly the same. So when you work with the Laplacian matrix, you are mostly interested in the smallest eigenvalues. And when you work with the adjacency matrix, you just focus on the largest eigenvalues, but it's exactly the same. So we denote by lambda1, lambda p, the p eigenvalues of the adjacency matrix, and v1, vp, the associated eigenvectors. And capital Vk will be the eigenspace generated by the k largest eigenvectors. So as I said, when you work with L, you are interested with the smallest one. When you work with the adjacency matrix, you are interested by this in this largest eigenvectors. For example, spectral clustering algorithm. No, if your graph is perfect, you don't have any perturbation, then this largest eigenvectors will give you the will directly give you the structure of the graph. So you can also write this problem as the following optimization problems. If you want to recover the first component, so we assume that the first component is the smallest one, So if you want to recover the first component, which is the smallest one, you just look for the sparsest here eigenvector. So if your graph is ideal, so there is no perturbation, it's just because the sparsest eigenvector is the one with the less number of coefficients, and obviously it's an indicator vector corresponding to the first connected component. So it's easy to see that it works well. If you want to recover the second component now, what you can do, exactly the same. In this eigenvector, in this eigenspace generated by the k largest eigenvectors, you are looking for not the sparsest one, but the second sparsest one. So it's still the sparsest one, which is orthogonal to the indicator vector corresponding to the first component. And you can do exactly the same for the third component, fourth component, etc., etc., etc. So if you want to recover this component here, you can solve this minimization problem. So you are looking for the eigenvector, the sparsest eigenvector, which is orthogonal to all previous connected components, and which is still in the eigenspace generated by the largest eigenvalues. So now if you're familiar with this L0 penalization problem, it's quite challenging. So it's an NPR problem due to the presence of this L0 norm. So what you usually do, you just replace the L0 norm by the L1 norm here. And of course, you want to show that these two problems here are equivalent. And you can show that these two problems are equivalent if you assume something else. So you have to assume that you know at least one element belonging to each component. So from now on, we assume that for the k component you want to recover or you want to find, you have to know at least one node belonging to this component. And I will denote by capital V tilde k, this space. So it's a space, eigenspace generated by the k largest eigenvectors. And with the assumption that for all that you know at least one node belonging to this component. And these nodes will be called representative elements. Okay, so now it should be easier to solve this kind of problem because you have L1 norm penalization problem, so it's quite easy, but you still have some problem because this space here of constraints is not so, I mean, easy to understand. So fortunately, you can rewrite this set of constraints using the fact that all the eigenvectors are orthogonal. So if you write your orthogonality constraints, you can show that this space here of constraints can be rewrite as the linear, as the equality constraints here, there. So we denote by U. So U will be the matrix formed by all other eigenvectors. So Vk was the eigenspace generated by the largest eigenvalues, and now we are focusing on the smallest eigenvalues. So we denote by Uk the eigenvectors associated with the smallest eigenvalues. We denote by W first row, capital W all other rows here, and you can simply rewrite here this space here like this. So it's exactly the same as solving capital W V equals minus W. Of course, with the constraints that you know your representative element belongs to this component. So now we assume that some of you know well that you can rewrite this penalization problem like this. Okay, so it's I call this problem P Lasso because it's very, I mean, it's it's like a Lasso problem. You have a L2 norm here on your capital WV plus W, and you have penalization, L1 penalization on the number of the solution V. So our algorithm looks like this. So as input, you have your graph G, you have the adjacency matrix A, you have the number of clusters to recover, and these assumptions that you know one node belonging to each component. So you need to have a family of elements which are all in the k-hat clusters to recover. So first you perform your spectral decomposition on A, you sort the eigenvalues and the eigenvectors V1, Vp. Sorry. And for each component you want to find, you first define UK as the first eigenvector, so the eigenvectors associated with the smallest eigenvalues. You split U into two parts, W, which is the first row of U, and all other rows, capital W. You solve the L1 minimization problem I've just shown you, P Lasso. When you solve the L1 minimization problem, you find the indicator of the first component. Of course, you have to add this representative element here. You update your matrix of eigenvectors here. So you just remove, for example, you have recovered the first component, so you remove V1 and you change V1 for this indicator of the first component. And you perform a Gram-Schmidt orthogonalization just to be sure that all these eigenvectors are still orthogonal. And you do the same for all components, and at the end, if it works, you can you recover your graph. So now I just want to give you an idea of how you can, for example, solve this L1 minimization problem, how you can find the number of clusters, and how you can, in practice, define a family of representative elements. So first, the L1 minimization problem. So it's not very complicated. So if you want to solve such a problem here, you have some solutions. So the first one, I will begin by the second point. The first one is to use Lasso or Lasso algorithm from type Shirani. So for example, you use the R package glmnet, and you will find a solution easily, the solution V star. So something very important is the calibration of the penalty here, lambda. So if you take a very large lambda, then you will penalize a lot your problem. So you will find a very sparse V. And it means that your cluster will be quite empty. And if you calibrate your penalization problem parameter is too small, then you won't penalize a lot your problem and your cluster will be very big. So you have to be very careful when you calibrate your penalization problem. And you will just calibrate it by using a 5-fold cross-validation. So the second solution is to use what is called hard thresholding. So what you can do, you can just solve the least square problem here. So you just remove the L1 norm, L1 penalty here, and you solve this problem. And given a threshold, you threshold your solution. So you just say, okay, all coefficients which are smaller than t are equal to 0, and 1 otherwise. And now it's very, nice to see why these two solutions are quite the same. So I have I have some graphs to show you. So here I took a graph and different perturbed version of the same graph, so with perturbation ranging from 0.01 to 0.5. And if you are familiar with the glmnet package and Lasso, etc., you will, it will ring a bell. So here you represent the evolution of all coefficients solutions of this problem according to some different values of lambda. So here the red coefficients correspond to you want to find, and the black one correspond to all other nodes. And what you can see, if the graph is not so perturbed, so here the two first graphs, if the graph is too not perturbed, the red line are very far away from the black one. So it means that if you use Lasso, so if, for example, you have a threshold on the L1 norm value here, so here is L1 norm value, you have your threshold on the L1 norm here. It's exactly the same as thresholding the coefficient of the solution here. So you still recover perfectly your components. And here it's exactly the same. So Lasso solution and hard thresholding solution are exactly the same. But when your graph become perturbed, it's quite harder. So here, for example, you can see that black lines are very close to red lines when you look at the L1 norm. So you have some coefficients here which enter the model before some red ones. So if you use Lasso or you threshold your coefficient, it won't work. You will make some mistake, and when the graph is perturbed, it doesn't work at all. Okay. So I already talked about how to define the number of clusters. So as I said, when your graph is not perturbed, you just look at the multiplicity of the eigenvalue, and you can do the same when the graph is perturbed. So here I represented the eigenvalues for different perturbation of the same graph. So in red here, you can see when the graph is not perturbed, here, for example, the eigenvalue 0 is of multiplicity 3. So it's the best, I mean, it's the number of connected components in the graph. And when the graph is not, I mean, is perturbed with a perturbation of 0.01, it's the orange line here, you can still see that the eigenvalues are not so far from 0, and you still see an eigengap here. So you can say, okay, the optimal number of connected components will be 3. When the graph is a little bit more perturbed, for example, 0.1 in light blue, here it's harder to find the optimal number of clusters because your eigenvalues are not so, I mean, there is no eigengap in your values, so it's harder to find the best number of connected components, 3. But we are still looking for an eigengap in the data. And to finish with this part, how you can find the representative elements. So when you work with real data, it can be a restrictive assumption, but when you work with some biologists, I mean, they always have some ideas of which genes are very interesting. So they can give you just a list of genes, and you try to find some clusters centered on these genes. But you can also use, for example, the betweenness centrality score to find ups in your graph, and then you cluster, I mean, you use your algorithm to find clusters centered around these ups. Okay, so I have time, so I can turn to the simulation part. So we made a lot of simulations to see how our algorithm behave. So first, I simulated 100 versions of the same perturbed graphs with different value of perturbations, so ranging from 0.01 to 0.5. So just because the main goal was to have, I mean, better results as the spectral algorithm, which does not work with large perturbations. And we also define some different settings. So for example, the number of nodes was from 50 to 1000. A number of connected components, which depends on the number of nodes. so with a ratio between 1% and 20%. The connected components were chosen in a balanced way. So just we would like to have the same number of nodes in all components. And we compared our algorithm with different methods. So the first class of method we compare with were non-self-tuned methods. So it's just what I call non-self-tuned method is just you need to give the number of connected components of the true graph. And so we compared with the spectral clustering algorithm as defined, as previously defined, and a regularized version of the spectral. And we also compare to self-tuned methods. So self-tuned methods, in contrast with non-self-tuned methods, are methods that do not require the true number of connected components to be run. And we compare with the hybrid algorithm from Combe et al. And the Markov clustering algorithm from Van Dongen. And the performances were measured using the Adjusted Mutual Information Score, AMI. So it's a score between 0 and 1, and the closer to 1, the better the method. And we chose this score because it allows the comparison of methods with a different number of clusters, which is exactly what we have. So I don't really like presenting such table because it's very, it's very hard to see something, but I just wanted to give you an idea of how algorithm behaves on such data with different values of the parameters. So you just need to know that if you increase the perturbation here, so you have, for example, 0.5, 0.5, so results are very bad. It's not normal. So if you don't have any perturbation, it works well. And if you increase the perturbation, it's quite hard. The second thing you need to know is that the number of nodes here from 50 to 1000 is not the main problem. I mean, even if you have more than 1000 of nodes, it still works very well, which is good. So it's not a big problem to increase the dimension of the problem. So the most important parameter is the number of connected components, K. So for example, here, if you have only 2 clusters, it's quite hard for the L1 spectral clustering to recover the connected components with an AMI of 0.5, even if the graph is not perturbed. So it's just because the L1 spectral clustering is a sparse method. So if we have to better calibrate the penalization problem. So we forgot lots of nodes and we don't have good performances. And at the opposite, if you have a very large number of clusters, for example, here, 100, it doesn't work. So here it's not a problem of sparsity, it's just a problem of you have too many clusters and it doesn't work at all. And at the opposite, if you have a good ratio between the number of connected components and the number of nodes, for example, here 2% or 1%, it works very well. So you have around an AMI of 1, and even when the graph is perturbed. So for example, here with a perturbation of 0.25, which is quite big, you have very good performances. Okay, so if we compare with the state of the art, so here you can see exactly the same settings that are already explained. So you have your perturbations here from 0.1 here to 0.5 here. So I remove the first line just because 0.01 is too easy. So here you have the results for around 100 of nodes and a number of connected components of 10. In orange, brown, beige, you can see the non-self-tuned method. So here you know the number of connected components, so it should be easier. And in blue, you can see the algorithm where you don't know the number of connected components. So what you first see is that here the orange algorithms or the orange performances are quite the same. So our algorithm is beige one here. So we have exactly the same performances. Sometimes we are a little bit worse and sometimes we are a little bit better. So I added some red diamonds. So these red diamonds indicate the number of misclassified nodes, so the number of errors we made. And something very interesting, because even if our performances are quite a little bit poor here, you can see that the number of misclassified nodes is smaller than for other methods. So just again, due to the fact that our algorithm is sparse, we prefer just forgetting some nodes. So we have worse AMI, but we still don't do mistakes. Okay, in blue, you can see the results for the self-tuned methods. And as you can see, our results are a little bit, I mean, we are a little bit poorer than the Markov clustering or sometimes the hybrid algorithm. But when the graph is perturbed, we are very competitive and we outperform those algorithms because they don't cluster anything. So they just put all nodes in the same cluster, so they don't work, and we are quite good. And we have the same results for 500 nodes and 10 clusters. So as you can see, it's easier. All methods are very good, except when the graph is perturbed. And again, we are better than other when the graph is perturbed. Okay, so I just have 2 minutes. So to finish, I just would like to give you an idea of how you can use this algorithm with the biological data. So I use data from the TCGA. So I don't know what if you know what the TCGA is. So it's a big American project from the NIH, and this project was about to characterize the cancer genome. So you have data from more than 20 cancer and 500 patients each. And you have gene expression data, copy number, mutation, pathology, medical images. So you can, if you want some data, you can just download. So it was before COVID. But now with COVID, we have lots of data. So here we downloaded some gene expression data with around 16,000 of genes and 500 of cancer patients. And we choose, I forgot to say that, but we focused on kidney cancer. So we applied the l1-spectral clustering to this kidney cancer data. So first we need a network to do some clustering. So I created a correlated network. So I measured the correlation between all genes, and I thresholded for correlation smaller than 0.7. And my network is made with around 5,000 of genes. And after applying the l1-spectral clustering, so we found this kind of network. So we have around 200 components with an average number of genes of 27. And we found five very interesting components. So I don't have time to explain what kind of biological analysis we could make, but I can just show you something. For example, the blue one is made of 38 genes. And we Googled all these genes and we found an arrangement in inflammatory response which could say, okay, these genes or this cluster of genes is related in some way with inflammatory response for patients which have kidney cancer. And to confirm this association, we just measure the correlation between the cluster expression. So it's the average expression of all genes belonging to this component, with two very famous genes involved in inflammatory response, and we found a significant correlation. And we also computed the correlation or the association between the expression of the cluster and the grade. So the grade is just how strong the cancer is for each patient, and we found a very strong association. And you can also do this kind of analysis. So for example, the yellow cluster here is made of around 100 of genes, and we found, doing some survival analysis, we found that if you have a high gene expression from the cluster here, it means that you have a significantly good survival compared to people with a low expression of the cluster. So of course we need to talk with the biologists to see whether it's interesting or whether they know these genes in the clusters, but it's still, I mean, it's very interesting to know that you can use this kind of algorithm to find some new things on cancer. So we just finished by the conclusion. So we developed an algorithm which seems to work quite well on simulated data, and which can be used on real data, but further work include how you can calibrate the optimal number of clusters and the representative elements, and also the penalty. And from a theoretical point of view, we also would like to show that some theoretical results of like those which were obtained for the spectral algorithm to validate the performances of our algorithm. Okay. So here are some references, and feel free to ask me a question if you would like to. Thanks.